every living organism on Earth has genetic information stored in deoxyribonucleic acid, or shorthand, the DNA. What is DNA composed of? Well, it'll be two chains. If it helps highlight it, this would be one chain. Here's the other. We're looking at just a slice of this. And along them, they'll be composed of cytosine, guanine, adenine, and thymine, all forming specific pairs. Adenine will always bond to a thymine, and guanine will always bond to a cytosine. So if you have one strand of DNA, if you have just one part of it, then you immediately know what the other half says, because A's always bond to T's, G's always bond to C's. And so you have DNA, when it's stored, is this double helix pattern. It'll be these two chains twisting and twirling on top of each other. Now, what does DNA do for us? Well, like we said, it's this genetic information. Its purpose? Well, when we read it, we can build proteins. So let's set up what we mean by what proteins are and what they do for us. Proteins are even bigger, complex, large molecules, and they are constructed out of long chains of amino acids. The amino acids are going to be very important for us when we start to get back towards more astronomical topics. But the emphasis here, these proteins are a big chain of amino acids that will then fold in on themselves into some sort of shape. And just like when we were talking about the shapes of carbon having different properties, the shape of protein determines their function. Shape is absolutely essential in understanding what these things do. What we have pictured here is myoglobin protein. And what this does is basically it's the oxygen carrying pigment. And this is how oxygen is being carried in your muscle tissue. And so this is one example, a type of protein that your body needs to function. And this protein is constructed from your DNA. So let's go through how our strands of DNA will build up these amino acids to form any protein that we need to function. Before I start these next few slides, I just want to put a little disclaimer out there, especially for all you biologists. I'm going to be glossing over a lot of fascinating, intricate details. I'm only going to focus on the primary steps that matter for us. So to the biologists out there listening to this, I apologize. We are just glossing over this. To you non-biologists, I strongly encourage you to delve deeper into this. There is fascinating depths of material that we are just touching on. So with that disclaimer out of the way, how do we go from DNA to protein? Well, our DNA is stored in the chromosomes, and you can imagine unwinding it, stretching out, and getting our DNA strands. And here you can see that double helix pattern that we're talking about, with the sequences of adenine, guanines, thymines, and cytosines. What we're going to see once I start this animation is a RNA polymerase glob onto the DNA, start to read it, and construct what we call a messenger RNA. So get the animation going, and here's the polymerase bonding to the DNA. What it does is it split up the chain and starts attaching the corresponding sequence, reading one half, and it will spit out, there's that messenger RNA. So I'll let it play one more time. The RNA polymerase globs onto the DNA and starts building, reading basically half of the DNA information. Because as the order the material is allowed to bond together, it's easy to construct the corresponding second half of the chain. I'll let it go one more time. Now, this new information, this is the messenger RNA. And that is what's going to exit out of the system. That will leave the system, exit out of the nucleus, and go on to the next step. So now that messenger RNA, this stuff, the strand that was read off of the DNA, now that that's outside of the nucleus, Yes, it can be globbed onto by a ribosome. And the ribosome is going to read the messenger RNA and start constructing a chain of amino acids. So let's go ahead and fire up this animation. So the ribosome grabs onto the messenger RNA and it starts attaching through transfer RNA amino acids. And that's some important things we talked about earlier. So the transfer RNA comes in these triplets of ATCGs, hence why we call this genetic coding a sequence. And that amino acid gets deposited on. One more time. Amino acid through the transfer RNA gets attached together, and the amino acid chain starts to grow and expand until finally that amino acid chain is released and then folds on itself due to its own chemical reactions, the physics pushing and pulling on that chain of amino acids, and it literally folds into the necessary protein that has been constructed. So, how do you go from DNA to proteins? As a reminder, you read off the genetic code, creating a messenger RNA. That messenger RNA gets read by ribosomes that will attach individual amino acids. And when it's done, the amino acid is released, and the amino acids will kink, contort, twist all onto itself until it's reached the necessary shape for the proteins that go on to do the necessary biological functions in the organism. And this is what we mean by DNA is genetic information. If you have the DNA, you can construct the necessary proteins. So let's keep upgrading, going from small stuff to bigger and bigger, and start pushing into what is alive. Here's an example of a prion. The one's pictured here is literally bovine prion protein, better known as is the source of mad cow disease. So, a prion is a protein that just misfolded onto itself. Something happened to that protein that it didn't fold into the correct shape. And a recurring theme, recall that if form determines function, if you did not fold in on properly on yourself, then your physical properties are different. Going beyond this, once you have a prion, a protein that misfolded, that prion can transmit that information of how to misfold to other healthy proteins. So we know that prions can be sources of neurological diseases. And so I ask myself, is this alive? Is this protein, all of its properties are determined by how it's contorted and twisted? Is that alive? My gut feeling is to say no, and I'll shortcut the suspense and tell you that the scientific community does not define prions as being living organisms. Let's try another one. Let's try viruses. So the term virus actually comes from Latin to refer to poison. And as we know now, viruses tend to be infectious agents. What they'll do is they'll attach to a host cell and just hijack that cell's ability to replicate itself. Here's a classic example of a virus. You've got the virus with its spider-like legs and this shell. And inside the shell is the virus DNA or RNA, depends on the specific virus you're dealing with. But either way, you've got this protein capsid surrounding the virus's genetic information. And so what this virus will do is grab onto a healthy cell and then just inject its viral information, its viral genes. Now this virus has hijacked the cell's normal function. Right? The cell's process of reading genetic code and producing the necessary proteins have been taken over, because now that cell is going to start reading the viral DNA, start producing virus proteins, and eventually create new viruses. So, is this alive? Does this meet that definition? Let's go into what the community defines to be alive. And in general, it boils down to these 7 ideas. In order to be alive, One of the aspects is that there has to be growth and development. You must be growing up, changing in size. That has to be an aspect of reproduction. Have to have offspring. In order to be alive, something about you must be passed on, and those traits, hereditary, this is our genetic information, is being passed on to your offspring, how you reproduce. Another aspect is we say you must achieve homeostasis. You're trying to reach stable inner conditions so that the biological processes happening inside your body keep happening. If all of a sudden you had a chemical reaction inside you that got too hot and you literally burned your gut out, well, you're not in homeostasis anymore. You'll just cease to function. So you need to have mechanisms in your body that brings you back to stasis. Keep going. We have a metabolism. We need to eat. We are constantly consuming energy and burning energy. Just the process of living is a burn of energy. Lifting your arm up translates some chemical energy into mechanical. And as you burn up energy, you need to replenish it. So that's what we mean by our metabolism. Continuing on the chain, you have our cellular structure composition. You're composed of smaller units that work together to form bigger, complex pieces. And lastly, you need to respond to the environment. Right? If you see a flashing headlights, you know to cease moving. If a microorganism feels that the water around it is becoming more acidic, then an organism needs to be able to detect the danger and swim away. So this is how we define life on this big scale. So coming back to that question about viruses, well, the viruses, they don't reproduce. Yes, you can create more viruses, but the virus doesn't do it itself. It just hijacks another organism's ability to reproduce. And so, with all these ideas in mind, the consensus is that viruses are not living organisms, but what we call inert chemicals. Things that are on the transition to becoming living organisms, but not quite there. By now, I hope you're starting to see how we've built complex organisms out of the simpler stuff. Start off with atoms, and those atoms start chaining together to make simple molecules. Those simple molecules start to become materials that function in a living organism. Start stitching those together into these macromolecules, polymer complexes like your muscle tissue. Your muscles are this chain of macromolecules working together. Well, now you have a bunch of muscle tissue and organs. You start to create metabolic networks. And through all that, you can have your living cells. And so life, like we said at the beginning, is this growing tree of complexity. Start simple, and let the simple grow out towards complex living things.